Well, I want to apologize. We've had a program committee, an electronic glitch here. So I'll be moderating this pipeline session. We will get everybody in. And it looks like we have five presentations. And it's now 3:45, so we should run right to about 5 o'clock as planned. And I again apologize to the speakers in the audience that we didn't get this set up quite perfectly. So the first speaker is here, and I understand the slides are available. Dr. Nishi? Okay. Dr. Nishi's talk is TAC-935, reduces seizure frequency and severity and prevents premature lethality in SC1A plus minus Dravet mice. Thank you very much. Here's the
this is pointer
right there. Yeah. Okay.
Thank you for introduction. And I am Toshiya Nishi. I'm preclinical pharmacologist. And thank you for the ASENT organization organizers granting me an opportunity to go over my presentation. I brought 2 poster presentations to ASENT, and this is one of them. Talking about the preclinical evaluation of TAC-935, a new investigational drug in Dravet syndrome mouse model. I'm going to talk about this presentation on behalf of the Jennifer Kunis and Nicole from Northwestern University team. So I am employed by Takeda Pharmaceuticals, and this study was funded by Takeda and Neurotherapeutics. These 2 companies are now currently jointly developing this investigational drug, TAC-935, for treatment of rare epilepsy, including Dravet syndrome and Lennox-Gastaut in the epileptic encephalopathy category. So allow me to spend a couple of minutes introducing you to what TAC-935 really is. This is totally new one, and the target is really unprecedented one, and I guess this has been nobody's radar yet. This is a small molecule specific inhibitor of enzyme known as cholesterol 24 hydroxylase. This is responsible for cholesterol catabolism in the brain, converting cholesterol into this metabolite 24 hydroxycholesterol. Just adding this hydrogen moiety here and adding hydrophilicity and helping this cholesterol metabolite to get excreted from the brain to the periphery and leading to the bile acid metabolism in the liver. So the TAC-935 inhibit this reaction. So we can measure this 24 hydroxycholesterol. This is a little bit too long, so let me call it 24HC. So we can measure this metabolite as a very specific pharmacodynamic marker indicative of enzyme inhibition by this small molecule. We can measure it certainly in the brain, only in animals, and we can't measure it in the brain in humans, but we can measure this in the plasma in humans because this goes through blood-brain barrier, and the 24C level in the plasma is purely almost 100% brain origin. So this will serve as a very ideal clinically monitorable pharmacodynamic target engagement biomarker. And this is important not only as a plasma biomarker, but also certainly as the bioactive metabolite in the brain, especially known for positive allosteric modulation of NMDA receptors, and also known for some other things like inflammation and cell death. And all these neurotoxic liabilities make sense in neurological disorders, including epilepsy. So with that said, we tested TAC-935 in a couple of different epilepsy models, And it worked in some of the models, but not all. I would say it has a unique pharmacological profile compared to other classic anti-epileptic drug. And in addition to that, we knew that inhibition of this enzyme brings about a very interesting survival benefits in Alzheimer's disease mouse model. So we naturally came to think that why don't we test TK935 in an epilepsy model with a high mortality? So we selected Dravet syndrome mouse model. So Dravet syndrome is a rare pediatric onset epilepsy characteristic to febrile seizure, as well as the high mortality due to unexpected seizure, known as SUDEP. From etiology viewpoint, around 80% of patients have a loss of function type of mutation in SCN1A gene coding NAV1.1 sodium channel. So this etiology can be reliably modeled in animals by genetically deleting this gene. And we worked together with Northwestern University, Jennifer Kearney, who developed one of the Dravet syndrome mouse model, which has a very interesting phenotypes, 3 key phenotypes linking directly to human symptoms. The first, the existence of spontaneous seizures. Second, the high mortality, SUDEP type phenotype. Also, the low threshold to hypothermia-induced seizures. And we tested 935 in this mouse model. So, like I said, this model has spontaneous seizures, but we need a little bit of priming stimulation to unmask their epileptic phenotype. Right after weaning, just a little bit of addition of hypothermia stimulation to promote spontaneous seizures. And that was followed by initiation of drug treatment, TK935, for 2 weeks. During that period, we monitored spontaneous seizure as well as mortality. And we wanted to also evaluate drug effects on the hypothermia-induced seizure threshold. So we need a separate cohort to evaluate that. So that's about the study design. So this is basically seizure diary of individual animals. As you can see clearly, the TK935 treatment almost eliminated spontaneous seizures, except for a couple of individuals, female individuals with high seizure frequencies. So if you could look at this note, the red triangle marks the death, SUDEP. So as you can see here, there are no red triangles, meaning the complete elimination of SUDEP in this Dravet syndrome mouse model. So like here, the TK935 treated arms showed no SUDEP in this model. This is really, really phenomenal. And I should point out here that the effects of seizure reduction in this mouse model do not necessarily mean the protection of mice from SUDEP. One example is lamotrigine, which actually worsens seizure frequency in this model, but lamotrigine protects mice from SUDEP. So there is an apparent disconnection between spontaneous seizure and the SUDEP in this model, which is a very interesting thing. But this compound can address these two elements. And we measured drug exposures and like I said, the brain level of a 24-HC enzyme metabolite. So this is just about 50% of enzyme inhibition, which is, I would say moderate, not complete inhibition, which was actually sufficient to bring about this level of efficacy in Dravet syndrome in this model. And last but not least, we measured hypothermia-induced seizure threshold, which is actually kind of hyper in this model. There are not so many drugs that can address three disease phenotypes in this model. The slightly but significantly TKNIV5 raised hypothermia seizure threshold for 1 week treatment. So that said, the TKNIV5 treatment addressed significantly 3 key disease phenotypes in this Dravet syndrome mouse model: reduction of spontaneous seizures, seizure frequency, and seizure-related mortality, and as well as the slight elevation of seizure threshold induced by hypothermia stimulation. So with that said, this study suggests the CH24H inhibition might hold a promise to a totally new treatment approach to rare type of disease, including Dravet syndrome. Thank you for listening. I'm happy to take any questions.
About toxicity of the compound, potential toxicity.
That's what I'm really interested in. But honestly speaking, I haven't seen any weird things in animals. I tested the longest treatment was 5 months, but no effects on no effects on obvious behaviors and body weight. No obvious toxicity so far.
The second question is, I noticed in your summary slide you referred to premature reduction in premature lethality, but you mentioned SUDEP, and I think that's one of the questions in the use of unexpected death in all kinds of mutant models, whether it really is SUDEP or whether it's just a process of ongoing degeneration, or whether it's a consequence of repeated seizures and short status. So do you have any sense of those distinctions in the animals that you were examining?
That's really wonderful question. And my another presentation is also about kind of SUDEP model. And one of the current hypotheses behind the SUDEP is the propagation of spreading depression into the brainstem region, and leading to the execution process of cardiac and respiratory arrest. And this Dravet syndrome mouse model is known for a lower threshold to spreading depression. So I suspect that pathology is contributing to the SUDEP phenotype in this mouse model.
Thanks very much. Can you tell us something about the genetic background of this line and whether it is a single inbred mouse line? And if that's the case, do you have any intentions in checking whether this is effective in other lines, given that we know that background genetics has a huge influence on the determination of outcome?
It does. And this is a hybrid. The genetic background is hybrid between 129 and C57. So C57 is known for, high susceptibility, the opposite, high susceptibility to seizures. So the 129 is a little bit milder. So, Jennifer Kearney blended to, genetic background to adjust, nice level of seizure susceptibility. So this is F1 hybrid background.
We have time for one more question.
Just briefly, you alluded to it, but you didn't say very much. Can you say anything about the specificity of this agent in Dravet's versus other epilepsy models, other situations? There's this whole trend, I think, right now to think about orphan development programs and find an orphan for which a drug has value and perhaps ignore any other place where it may have value. What do you know about that?
Right now, that's a really good question. I have no immediate answer, but I think there are no reason to specify the drug effects on Dravet syndrome specifically. But because of the lack of animal models for other rare epilepsy models, I haven't tested this compound in other epileptic encephalopathy models.
Thank you very much.
Thank you.
The next paper is longitudinal 148-week extension of Anavex 273 for the treatment of Alzheimer's, demonstrates maintained activities of daily living scores, reduced cognitive decline for patient cohort on a higher drug concentration, and confirms the role of precision medicine patient selection biomarkers by Walter Kaufman at Anavex. Thank you, Walter.
Yeah. Hello. I hope that you are not intimidated by this title I didn't choose. It's very complicated. In terms of disclosure, I am the Chief Medical Officer of Anavex Life Sciences. So what I'm going to present today is an example of the approach that we are taking to Alzheimer's disease that can be extended to other of the studies that we are conducting with these type of drugs. What are we working on? I'm sorry, yes, I have a difficulty. I'm talking about sigma-1 receptor agonist. The rationale for this is had been demonstrated through now quite extensive literature showing that this non-opioid sigma-1 receptor has an important role in homeostasis and is key for the interaction between the endoplasmic reticulum and mitochondria. And specifically in the case of neurodegenerative disorders, has a dual set of actions. One, as you can see at the center, I don't know if I can, oh, I'm sorry, this is not working. Reference at the center shows that can decrease or ameliorate the underlying processes in Alzheimer's disease, in this case, tau hyperphosphorylation, but also has a role in I'm sorry, thanks, Tom. Role in compensatory phenomena. And there is an example of areas in which the sigma-1 receptor activity is important are mitochondrial function, autophagy, neuroinflammation, etc. So the data that I'm going to present today relate to predictors of outcome, specifically of trajectory of these two key outcome measures of cognitive function and adaptive function in Alzheimer's disease. One is the mini-mental state examination, and the other one is an adaptive skills, activities of daily living skills developed by an Alzheimer's disease consortium. So the data come from two studies. The first one is a phase 2A study that had two components. The primary goal was to determine safety and tolerability of the drug, but also to gain information about efficacy. This study that lasted 57 weeks extended into an extension study has lasted additional 208 weeks. The data that contributed to this result that I'm going to present has to do with the first study, the one with part 1 and part A and part B, as well as with the first section, the 2/3 or so of the extension study up to week 148. And as an open label study, what we can look at is trajectories, whether there are different trajectories among the patients, as well as comparison with what is known about the decline of these cognitive and functional markers. There is very good data on national history in Alzheimer's disease. So the first is the concentration of the drug. This is a sigma-1 receptor agonist that has also muscarinic receptor modulation function, mainly agonist, but not exclusive agonist. So the first thing is, what about concentration? And when you look at concentration average throughout the Part B, the main component of the phase 2A study, what you see is that concentration has an effect on the scores, on the delta scores, how much the score changed from baseline. If there is decline, you will have a negative delta, and the closer to zero or a positive means that cognitive function or adaptive function had been preserved. As you can see in this slide, when you group the patients, regardless of the dose, but you group them in terms of concentration, what you see is that there are essentially two different profiles. Those with high concentration have a delta that is pretty much preserved when compared with those with low and medium concentration. So when you plot this in a different way, now in terms of the trajectories, you see a clear separation between those that have high concentration and those had either a low or medium concentration. It's quite dramatic, and this is on the adapted daily living skill scale. And we have introduced as covariates to these models the sigma-1 receptor genotype as well as cathecol orthometyl transfer genotype. I'm going to refer to sigma-1 receptor genotype also as a predictor of response later. Here you see the same situation, a little less dramatic with the mini-mental score. Also, there is less decline with higher concentrations of the drug. Now, I think one of the most interesting things, because it had to do with what precision or personalized medicine is, what happened with genomic biomarkers. And I'm going to present only part of the data we have. These data are focused on the main target of the drug. And this is important because this can be applied beyond this particular indication for Alzheimer's disease, but could be applied to any other condition in which you think of enhancing homeostasis. If this is drug specific. There are a handful of sequence variants of the sigma-1 receptor that have been described, and they seem to have a role in the function of the gene. And one of them in particular seemed to be very informative. This variant that affects the transmembrane domain is specifically an exon 2, and this protein that has, among other functions, being a chaperone, seems to be impaired in terms of trafficking. It's a single amino acid substitution from a hydrophilic glutamine to a lipophilic proline, which changes the configuration of the secondary structure of the protein. So what happened when individual had this sequence variant, which affects approximately 20% of the population? They have less preservation of either the MMSE or the activity of daily living skills. Either one of them is less preserved over time when you compare those with the variant versus those with the wild type. And this variant actually has function, an impact on a variety of neurology function. There's a recent paper published at the beginning of the year showing that there is the decreased sensitivity to pain in those individuals who carry this particular variant, particularly to pain induced by thermal factors or stimulation. Now, expanding the importance of the role of looking at the specific gene that is being targeted is the levels of mRNA, which also were measured, and this on the extension study, and it shows that when you divide the cohort between those that have a relatively stable delta or positive versus those that have a decline, those who have a decline, who have a negative delta or negative slope, they have lower levels of mRNA for the sigma-1 receptor. So it's clearly there is an effect of the gene target. I wrote in red so I don't forget that we're looking at other predictors, and there is later in the year there's going to be a presentation at the meeting about gut microbiome or microbiota, which is a very hot topic these days. So I wanted to conclude that this type of a study is, if you think is applicable to any other indication for which we're examining this drug, Anavex 273, this sigma-1 receptor agonist with also muscarinic receptor actions. These are ongoing clinical studies, and there is all of these are placebo-controlled studies and ongoing right now, one on Parkinson's disease dementia, the other one on Alzheimer's disease, a much larger study, and one that we just started a few weeks ago on Rett syndrome. And here is an sort of overview of them, and you can see the length of the study and the number of patients. The much larger studies, the Rett syndrome study will be followed by other larger studies is at the beginning, but you see that now we're expanding these through this study that I mentioned. The one that I had reported on is the phase 2A and its extension that is the fourth one. And so that's what I wanted to mention today, and I will be glad to entertain any questions. Thank you.
Questions for Dr. Kaufman? Sounds like it's all clear, Walter.
Amazing.
Thank you very much.
Thanks.
The next presentation is entitled Pharmacokinetics of Clobazam and N-desmethylclobazam following administration of clobazam oral film by Dr. Heller of the Pharma Study Design, LLC.
Thank you. First thing I want to explain is why the title of this is not as in the program. We thank the organizers of this meeting for having accepted 2 posters from Aquestive and also invited us to give a platform presentation where the suggestion was that we talk about both posters. And considering the length of time, we decided that we would choose one, but it's not the one in the program. So we're going to be talking about diazepam rather than clobazam. But I'd like to share with you that the 2 posters are posters 13 and 14. They're in the Brookside room, and we'd be happy to tell you about the content of both. Also, the clobazam work has since been published. It's most of the information in the poster is in a publication in the November issue of Epilepsia. So we're going to talk about diazepam, and this is also one of our posters, and a study comparing the performance of a diazepam film under periictal and interictal conditions. And just briefly to go through some background, seizure clusters commonly occur in patients with drug-refractory epilepsy. Classically, the mainstay of therapy is benzodiazepines, frequently diazepam. Now, diazepam buccal film, developed by Aquestive, is a novel dosage form with potential advantages. This is a postage stamp-sized film, a polymer, that adheres to the inner cheek, to the buccal mucosa. A portion of the drug is absorbed directly transmucosally, and we know that a portion of it is also swallowed and absorbed through the GI tract. The intended indication is selected patients with refractory epilepsy that require the intermittent use of diazepam. And this is, to my understanding, the only drug that has this indication right now is the diazepam rectal gel. So you can see that in principle, this dosage form would have some advantages over the standard of care. The study that I'm going to present is in adult subjects with epilepsy, and the objective was to assess whether dosing with this film at the time of a seizure was associated with any alteration in the systemic exposure as compared to when these patients are not having seizures. So very briefly, the design was a single-dose crossover in which subjects were monitored and PK samples were drawn during a epilepsy monitoring unit evaluation. They typically had seizures during this EMU admission, and we call that treatment B. And the second arm of the crossover was a visit to a clinic facility where PK samples were taken, but the intention was to obtain a pharmacokinetic profile under conditions, interictal conditions, when they were not having seizures. Treatment B, this is in the EMU associated with the seizure, and 12.5 milligrams were administered either during the observed seizure or within 5 minutes after cessation of the seizure. Treatment A, same dose, administered by definition, they had to be seizure-free for at least 3 hours. So that was the interictal condition. And the treatment could occur in either order. The subjects were adults, age 17 to 65, and the qualifying seizures had to be either tonic-clonic seizures or focal seizures with impaired awareness. The intended enrollment number was 35, and we wanted and enrolled subjects with fairly frequent seizures, as you can see. All of the subjects had to be on a background treatment of at least one AED, and most of them were on multiple AEDs. PK samples were collected at times shown, and during treatment B, which was the seizure treatment, samples were collected out until 4 hours. In the PK-only admission, the interictal, we collected samples out until 10 days. And we're going to focus mainly on the earlier part of the profile, but we were interested in variables such as clearance and so on. And obviously, safety and tolerability were evaluated throughout. At the end, 35 subjects received at least one dose of study drug, which is the safety population for this study. There were 21 of these who had complete PK data available for both treatments, extending the examination out to 2 hours post-treatment, and 16 with complete data out to 4 hours. And as you'll see from the footnote, there's a number of reasons that subjects did not qualify for the primary analysis. In some cases, subjects included in the safety population received only one of the treatments but didn't receive both. Occasionally, critical PK samples were missed. In some instances, pre-dose concentrations were elevated. And in one case, one of the investigators administered the drug, the film, contrary to the way it's recommended. So going to the results, which I'll show you in two ways. First, this average plasma concentration curve, and then we'll look specifically at the AUC and Cmax values. And you can see this is again for the 21 subjects who had valid PK data for both treatments A and B. This is simply a mean concentration curve, and the error bars are standard errors of the mean. The curve that appears to be at least numerically higher is the treatment B condition, the periictal ictal curve. And here are the actual data for analysis. And what we've shown here is the comparison for those who had 2-hour profiles above and 4-hour profiles below. And these are geometric mean values for the Cmax and for the partial AUC out to 2 hours or 4 hours respectively. And the only point here is, well, the third column is the ratio of the geometric means. And you can see they're not too different from one, and the confidence intervals are fairly well behaved. Clearly, there was no statistical difference. And we've also shown you Tmax, and Tmax by the Wilcoxon was not different either. So we concluded based on these data that a single dose of 12.5 milligrams administered as the diazepam buccal film provided exposure to diazepam under ictal periictal conditions that was comparable to that obtained under interictal conditions. And also that the diazepam film at 12.5 milligrams was safe and well tolerated. The most common AEs that were classified as either possibly or probably drug-related were somnolence and dizziness, which appeared at fairly low frequency. So I would be happy to answer any questions. Thank you.
Please identify yourselves for the audience when you have questions. Thanks.
So I'm Rod Scott from the University of Vermont and University College London. As I'm sure you know, buccal midazolam, liquid preparations of buccal midazolam are now the standard of care after being used for nearly 20 years in Europe and Australia for acute seizures in all sorts of acute settings. So my question for you is, why diazepam and why the film? It feels like there's a lot of reinvention of the wheel going on here, when in Europe this is completely standard.
Yeah. I don't know that I can answer that directly in terms of buccal midazolam, to my understanding, is not approved here in the U.S.
Correct.
Yeah.
I just wonder why there's been a move to diazepam in films instead of saying buccal midazolam. There's a lot of data. Why not try and get buccal midazolam approved in the United States? Why reinvent the wheel with a different benzodiazepine and a different administration?
Right. Well, I don't know if it's reinventing the wheel, but I think you raised the question as to why buccal midazolam is not approved in the U.S. And I don't know, do you?
So my understanding is it's entirely about being able to get intellectual property and being able to get a patent. So the midazolam we used 20 years ago was Versed. So when I first did the buccal midazolam experiments in London, we just took off the shelf Versed and put it in a syringe and put it into people's mouths.
Yeah.
That never got a patent. So the way that it went through the regulatory authorities in Europe was through some system that said if you have ongoing use for 10 years, then that counts as a trial, we'll give you a limited license. So there was a process in the European system to allow that. So my understanding in the United States is people want a patent in order to proceed, so they can make money, which is fair. I don't have a problem with that. It just seems that it's a very difficult way of going about doing something that exists in other parts of the world as standard of care.
Yeah, I don't fully understand the business aspects in terms of pursuing buccal midazolam, but I think it's fair to say that the standard of care at this moment is rectal diazepam. So I think you'd agree that there is some advantage.
That's what's licensed. I agree that's what's licensed.
So I think there's an advantage there.
What I actually use as a pediatric neurologist is buccal midazolam.
Right, right.
Yeah, I don't know if there's any FDA people here, but I'd have to imagine that a full process of data presentation and approval would be required in North America.
For sure. Even
for buccal midazolam. And I would also speculate that there's some kind of financial and issues about investment and return on the market, but speculation. Certainly you couldn't move to that without going through the kind of regulatory data acquisition that you're pursuing.
Yep.
No further questions. Thank you very much.
Thank you.
So the next paper it is long-term safety and tolerability of apomorphine sublingual film in patients with Parkinson's disease and off episodes by Dr. William Ondo, Methodist Neurological Institute.
Thank you. I'd like to thank the organizers for the invitation. These are my disclosures. I am a site investigator for the ongoing open-label safety study of this preparation of apomorphine. I'd like to spend about 1 minute on Parkinson's and maybe 2 minutes on apomorphine in general, and then discuss the specific adverse event profile for this product. Parkinson's, of course, is a very common neurodegenerative disorder of both the peripheral nervous system and the central nervous system. When you lose about 40% of dopaminergic neurons, you first develop the cardinal features of Parkinson's disease. Initially, the gold standard of treatment is levodopa, which is a short-acting precursor of dopamine. This initially is fairly effective. Despite the very, very short half-life of about 90 minutes of this drug, you often get 4 to 6 hours of duration of benefit. And it's thought that that is because the drug is taken up presynaptically into the remaining dopaminergic neurons and then released in a physiologic manner. However, as the disease progresses, you eventually lose at least 99% of dopaminergic neurons, and you're no longer able to buffer this system so that the duration of benefit shortens and starts to mimic the pharmacokinetics of the drug, resulting in these fluctuations that are shown here. Also, since you don't have any buffering capacity when you take a large dose of exogenous dopamine, you end up with these dyskinesias, the opposite of Parkinson's disease, where you move too much. And this makes levodopa, although still effective at treating symptoms, much, much more difficult to use. Now, apomorphine is a very interesting drug. It is an extremely potent lipophilic D1 and D2 agonist. The affinity profile pretty much mimics dopamine quite well. I make the point here, for those of you not familiar with this drug, it has no affinity for opioid receptors, despite the name apomorphine, so this is not contributing to the opioid epidemic. And in fact, the medication in the 60s and 70s was used to treat people from opioid withdrawal. If anyone ever managed to get through the whole book, Naked Lunch, William S. Burroughs used this medicine. He talked about the apomorphine cure to get off heroin. So there's some potential pathophysiology from that. It has a very long history. This arguably may be the oldest purely synthesized drug still in use, being developed in the 1860s. And it was in the 1950s and 1960s that it was first used, discovered that it was beneficial for patients with Parkinson's disease. The reason it never became a mainstay of therapy is because the drug itself has essentially zero oral bioavailability, undergoing extensive first-pass metabolism, so there's really no pill of apomorphine that's ever been practical. There have been a number of other preparations developed over the years. In the United States, currently, there is an injectable form of apomorphine, which is FDA approved for rescue therapy for people having off episodes, but it has never become extremely popular, I think largely because of the fact that it's an injection. Okay? There have been other attempts at sublingual and nasal apomorphine preparations, but they have generally been limited by significant irritation of the mucosa. All right, so this preparation, which is owned by Synovian, is a novel sublingual film of apomorphine. It has a lipid bilayer that buffers the acidic pH in order to try to mitigate the irritation that you get in mucosa from this medication. This data was actually presented yesterday by Dr. Factor, and I'm not going to go over it. I just wanted to make the point that in this phase 3 trial, there was an open label period where patients underwent titration of 6 different potential doses, and then a placebo-controlled period where they actually showed the significant efficacy compared to placebo. So we're going to go with the adverse event profile initially of this study first, okay, this 12-week study. So on your left is the open label portion for the titration, and you'll see these same adverse events pop up in various slides. This is the controlled portion of drug versus placebo, and you can see nausea is clearly the most common adverse event with this medication. Somnolence, dizziness, next most common. In this study, looking specifically at some of the other issues with the tolerability in the mucosa, you can see that you do have some irritation, dry mouth, some pain in the tongue, some swelling and edema. These numbers are all percentages, by the way, so 4% versus no percent in placebo. So that's the adverse event profile from the phase 3 trial. What I'd like to do is present additional safety data from the entire cohort of patients that have taken this medication. 3 studies which are completed, including the phase 3 trial I just discussed. This was essentially a single-dose QT study, and the only ongoing trial is the one we're participated in currently, which is this long-duration open-label safety study. So in total, 408 patients have received at least a single dose of apomorphine, but many of them only received a single dose. So we have 285 patients that were on it for some period of sustained time. So if you look at all these open label adverse events in the patients that took even a single dose, nausea is still the most common, dizzy, somnolence, yawning. Now, for those of you that have seen apomorphine, it's kind of interesting. Very, very dramatic medication because it's so rapidly acting. But what very commonly happens is the patient is off, they're stiff, they're having tremor, there's a big yawn, and then they come out of that yawn, they're on and fluid. It's really quite dramatic when you've seen cases of this. So it's sometimes, I think, difficult to differentiate the yawning and the somnolence, but you'll see yawning specifically pop in it because of that incidence. And I would argue it's probably a little bit more common than 11%. I just think it maybe is not often thought to be an adverse event as a single yawn. In the next population of 285 that have taken multiple doses, same pattern, okay, nausea, somnolence, dizziness, still the most common adverse events. Okay, now, we wanted to look at specifically patients that have taken for a longer period of time. So this is the cohort of the final study. The majority of these patients here have taken it between 6 and 9 months at the time of this data cut. A small percentage of patients have taken it more than 9 months. And you see the demographics here, fairly typical for a fluctuating Parkinson's disease trial. So the gross breakdown of adverse events in this long-term trial. So any drug-related adverse event ought to be about 54% of people. The serious adverse events for 4%, so about 3 people. None of these were really thought to be related to drug. One was a fractured leg, one was prostate cancer. So nothing that was thought to be related. Discontinuation adverse events, so a single patient discontinued here due to dental caries and then essentially gingival irritation, and no adverse events associated with death in the trial. If you want to break down the actual adverse events, again, this is its long-term use, specifically in patients that have been on for more than 6 months now. The same general pattern that you come up with, nausea. Again, this is an advanced population of Parkinson's, so fall is not too unexpected. And again, then you start to get this irritation, erythema of some sort in the mucosa, still 9% in this population. Again, and other things that would be expected in a long-term study like back pain. Okay, specifically looking at the tolerability, the oral tolerability of the medication in patients taking it greater than 6 months, you see again, overall erythema or redness in the gingival mucosa, the most common in 9%. You do get some patients that have some pain, dry mouth, some edema, 4%, so I guess that's about 3 patients that have had this. So overall, certainly compared to previous preparations, I think these are relatively good for chronic therapy, but there still is some irritation on the mucosa with the medication. Broken down into category, again, the same population, now greater than 6 months of use. Falls and injuries, still, again, 11% stands out, but not too surprising in this advanced Parkinson population. Somnolence and insomnia both reporting, again, very common adverse event in Parkinson's disease. The dizziness, hypotension. Hypotension is surprisingly absent in this because this drug is known to drop blood pressure acutely. And some of that may be dizziness in these reports, but relatively low numbers compared to other apomorphine preparations. And then again, hallucination, psychosis. And there were a couple episodes of syncope, 2 patients, I think. So in conclusion, the long-term data really showed similar adverse event profile to the phase 3 trial on shorter-term data. Most of the adverse events were either dopaminergic or irritation of the gingiva for the sublingual preparation. But overall, I think the medication was fairly well tolerated. I'd like to acknowledge the sponsors. Okay, Synovium has this, the patients that were in the study, and a variety of other groups, all the investigational sites that participated. Thank you.
Questions? So the numbers look good. Do people like it?
People like it. Doesn't taste too bad, okay, because I've been involved with a few sublingual apomorphine preparations over the years, and that was a complaint with some of them. It consistently works. The issue with apomorphine in general becomes, the utility overall compared to levodopa. It kicks in a little fast. There's not a head-to-head data, but it seems to kick in faster than levodopa, but is 15 minutes faster? How important is that overall utility? So the main reason I think people that drop it over time is not so much adverse events or lack of efficacy. It's just, is it worth the extra medication?
Thank you very much. Our last presentation is neuroprotection by endovascular selective cerebral hypothermia, experimental in vivo studies and initial clinical experience. It will be presented by Dr. Ron Soler, Renaissance Biomedical. Thank you very much.
Well, thank you very much. I'd like to disclose that I am a consultant and shareholder for Thermopeutics, and I'll be discussing their product this afternoon. Stroke is not death, to quote my colleague Eberhard Grube. And I know this to be true because my father had a stroke. It's not death, it's much, much worse. Globally, 1 of 4 people over the age of 25 will have a stroke in their lifetime, and nearly 2/3 will die or be disabled. The challenge in ischemic stroke is that there are many pathways that lead from occlusion to cell death. And while we want to open up that vessel as soon as possible, reestablishment of the blood flow can lead to further cell death by processes we commonly and collectively refer to as reperfusion injury. The latest advancements in acute stroke care center around reperfusion strategies. These are drugs such as lytics and devices, mechanical thrombectomy devices, that open up these large vessel occlusions. However, due to anatomical, physiological, and access to care issues, these latest advancements are really only been limited to about 8% of the patients. And in the patients that do get these procedures, because of reperfusion injury, only about half of them will improve and have no neurological problems, and they'll be functionally independent. So protecting the brain from ischemia and reperfusion injury today still remains to be one of the most important unmet medical needs. In the laboratory, therapeutic hypothermia has been shown to be the most robust neuroprotectant known to date because it acts on virtually all these pathways, and its role in mitigating reperfusion injury is impressively important. Unfortunately, in the clinical setting, in clinical trials, the techniques that have been used to induce hypothermia simply haven't worked. These are techniques that involve surface cooling or intravenous heat exchange, and what they do is they cool the whole body so that the blood that's going to flow to the brain gets cold, and then it's cold blood flowing to the brain that actually cools the brain. Now, these techniques are very slow, they're very unpredictable in actually cooling the brain, but more importantly, because of very serious detrimental systemic effects, and in particular cardiac dysfunction, they're limited to 32 to 33 degrees centigrade. However, if you look to the literature, the experimental literature, We know that time and temperature are the most important variables of therapeutic hypothermia and stroke. And there even may be an efficacy around 30 degrees. So this would explain why there's a discrepancy between bench and bedside with the clinical trials. To address these limitations, we developed a novel endovascular selective cerebral hypothermia device and technique. We call it TwinFlow. And TwinFlow provides very deep, rapid, and selective brain cooling, and it can cool faster and at temperatures much lower than any other hypothermia device. The catheter is a proprietary perfusion catheter that isolates and selectively perfuses the carotid artery with cold blood. It's used with any standard extracorporeal circuit. However, the setup is much simplified since membrane oxygenation is not required. The catheter is introduced through standard transfemoral percutaneous technique that's familiar to all interventional specialists. Under fluoroscopic control, it's advanced through the carotid artery. As shown here, a port located in the aortic arch receives the patient's warm blood. It's directed to the extracorporeal circuit, where the blood is then cooled and then pumped directly beyond a very atraumatic occlusion balloon positioned in the carotid artery, so that we essentially take over the blood flow to the brain with cold blood. This unique design has a countercurrent flow of blood where the cold blood goes in and the warm blood comes out. And this very effectively insulates the systemic circulation from the cold blood as it travels to the brain. In addition to providing cerebral hypothermia, the design also allows selective delivery of drugs and other devices right through the perfusion lumen while you are cooling. So if you're using in a procedure where you're using, say, lytics or mechanical thrombectomy, you can affect recanalization in a cooled, protected brain to avoid reperfusion injury. Now, when we use this in this application, we like to think we're providing intravascular cooling enhanced therapy. We're enhancing the recanalization. And so the company has taken the acronym and actually trademarked the iced tea. So this we're doing is serving the patient's iced tea. TwinFlow offers a very unique treatment paradigm in acute stroke so that it greatly has the potential to greatly increase the patient access to care. Since percutaneous carotid intervention is familiar to most interventional specialists, patient access is greatly achieved beyond what is currently available in by neuroendovascular specialists and centers today. And additionally, experimental and preliminary clinical studies suggest that hypothermia may also have similar neuroprotective effects in hemorrhagic stroke, so that this technology could be used in virtually all strokes. In vivo studies were carried out in large pigs, 50 to 70 kilogram pigs, and here we demonstrated we could very rapidly cool the brain to as low as 15 degrees centigrade without meaningfully affecting the systemic temperature. By simply controlling the flow rate of the blood and the perfusing temperature of the blood, we can have cooling rates as fast as 2 degrees centigrade per minute. We found that there were no adverse events, and systemic values, hemodynamic values, remained in normal ranges. And also, unlike systemic hypothermia, by simply stopping the cooling and allowing the brain to rewarm passively, we saw no rebound hyperthermia, which is very important because this could save us at least a day in the ICU. The catheter was studied in a very unique stroke model in pigs that was developed by Dr. Steve Lowney and his group up in London, Ontario. And in this model, one of the middle cerebral arteries was clipped surgically, and this was done for a period of 3 hours of ischemia. And then the clip was removed and followed by a 3-hour period of reperfusion, during which 25 pigs were randomly assigned to either selective cooling or serving as a normothermic control. At sacrifice, the brains were then examined by MRI and histology by experts who were blinded to the treatment groups. And what we found was there was no difference in any of the hemodynamic or systemic variables. However, there was a statistically significant reduction in stroke volume by cooling the brains to a mean 26.5 degrees for only 1 to 3 hours. Our initial human experience with the catheter has been in the setting of neurosurgery and refractory cardiac arrest. In this first patient, it was a 59-year-old man that had a giant aneurysm and needed surgical repair, and because of the long projected occlusion time, neuroprotection was recommended. In this case, the surgeon, who was familiar with using the catheter in the pig studies, used a twin flow catheter to cool the patient's brain to 26 degrees. And at the same time, systemic temperature was maintained at normal temperatures, demonstrating the very effective insulative properties of the system. The surgeon noted that the patient's recovery with no neurological deficit was much quicker than typically seen in this clinical setting. In a more dramatic case, this 38-year-old man arrested, with a non-shockable rhythm and had no flow for at least 8 minutes. The patient had no response to 44 minutes of manual CPR, and then 10 hours of extracorporeal membrane oxygenation at 33 degrees. So as a very last resort, twin flow was used to see what we could do with this patient. The patient's brain was cooled to around 22 to 25 degrees. We used a nasal probe as a surrogate temperature, and the nasal probe was at 27 degrees. Cooling was for around 12 hours. The patient made a full and complete recovery with no neurological deficit. And what was remarkable in this case was the patient was a urologist, and 2 months after his arrest, he was back at work performing Da Vinci robotic surgery. So in conclusion, hypothermia we know can protect the brain. We know that time and temperature are the most important parameters in hypothermia. And a catheter-based rapid, selective, and deep hypothermia technique may offer a means for improved clinical outcomes. Thank you very much.
So could you go into a little more detail about the mechanical features of the catheter?
It's actually quite simplified. It's a perfusion system. But instead of typical, for example, if you're familiar with bypass, where you take out of one perfusion catheter, you take venous blood, you hook it to the pump, you oxygenate it, you connect that to an atrial catheter, and you put that into an arterial catheter, and you put that in the artery, and you do a bypass that way. We have a single coaxial design. So through one catheter, we have the catheter position in the outer portion is in the aortic arch, where the blood comes out, and then it's pumped back in through an inner catheter. And it's this countercurrent flow of the warm blood on the outside of the inner tube, cold blood going inside, which provides insulation. So essentially all we're doing is we're perfusing cold blood to the brain. We have an occlusion balloon in the carotid so that we're just, we block the warm blood going in, only have cold blood. And we've learned that by perfusing at a pressure that's slightly above or at mean arterial pressure, we can cool both hemispheres from a single side.
So the heat transfer is direct right within the catheter?
That's right, yes. Mm-hmm. It's an active installation.
And I'm also just curious what stage of FDA device
Well, it's interesting.
regulation are you?
The device itself is FDA cleared as a perfusion catheter because using the 510K process, it's substantially equivalent to other perfusion catheters that are on the market. We do not have labeling for stroke, cardiac arrest, or any clinical indication, and so we've decided we're not going to go out there and just market it until we get some clinical studies in stroke, in cardiac arrest. Right now, there's a study going on in Taiwan. I showed you the first patient of that study. It's for refractory cardiac arrest, where it's being combined in ECMO in a really difficult patient population. And the first group of patients was presented last year at the American Heart Meeting, and the outcomes were quite remarkable compared to the center's experience with just ECMO alone in those patients.
To follow up on Dr. Satula's question, so if I understand, the blood goes through the sleeve, the outer catheter, comes out of the body, is cooled extracorporeally, and then goes back in cold through the core of the catheter. So does anything else happen to it while it's out? Does it get hyperoxygenated, or does its oxygen saturation stay stable? How's it treated when it's sort of being cooled, and how quickly is it cooled? Is it like a flash thing, or is it slow? What are the mechanics of this?
The mechanics of cooling are what we've been doing for almost 50 years with extracorporeal circulation and during a normal cardiac bypass. So we're using the same type of equipment. We even looked at, gee, can this be simplified? And we did a number of pig studies where we took a hemodialysis pump, a bucket of ice, and a metal heat exchanger. And we just flowed through it. The flow rates we typically run anywhere from as high as maybe 250 milliliters a minute down to as low as 100 milliliters a minute. And with that flow rate, the blood is cooled very, very rapidly. And we've cooled the blood. We've looked at temperatures as low as cooling the blood to 5 degrees and as warm as 20 degrees. And we found we could cool either way. It's just that it's the temperature of blood affects how fast you can cool. So we're still trying to figure out what's the best for these patients. You know, do you really overcool the blood, which is problematic because the blood viscosity increases, or do you keep it a little bit warmer but run your flow rate a little faster? And we found we can control this, which is very nice. One thing we did in all of our pigs, even when we were cooling the blood to 5 degrees centigrade, we saw no hemolysis. We were always looking for this. So we saw no problems of damaging the blood. So we're still trying to figure out the best ways of doing this, and we've kind of gone as far as we can in the animal models, and so now we're looking at putting together some clinical protocols.
It's very interesting, and if there's no further questions, thank you very much. That concludes this session. I want to again apologize to speakers in the audience for the slow start, and I'm glad we got it done on time, and thanks to all of you for a very interesting series of presentations. The poster reception and cocktail hour are underway.